Bonjour tout le monde, bienvenue à la présentation. Une présentation aujourd'hui de conférence conjointe CEF et IBIS. Donc, je me présente, Marc Mazereux, je suis directeur du Centre d'études de la forêt Université Laval et également membre associé de l'IBIS. J'ai un petit peu rapport quand même ici. Euh, Aujourd'hui, on a le plaisir d'accueillir Ilga Porth, qui est une prof de mon département, du département des sciences du bois et de la forêt, qui est arrivée à l'Université Laval en 2015, en même temps que moi, la même année. On s'est croisés à l'automne 2015. Je me rappelle, Ilga arrivait d'un postdoc de UBC, puis elle a fait auparavant, elle avait fait un doctorat à Vienne. Aujourd'hui, Ilga travaille sur plusieurs projets en génomique, une experte en génomique, roule un gros labo, plusieurs très prolifiques, plusieurs belles publications. Puis aujourd'hui, elle va nous parler des interactions plantes et herbivores, différentes applications. Fait que je t'invite maintenant, Ilga, à nous présenter ta conférence.
Ça fonctionne. Vous m'entendez? Allô? Ah, OK. Allô? Merci beaucoup pour l'appui. Merci, Marc, pour m'introduire. Je vous présente aujourd'hui certaines parts des travaux de mon laboratoire. Donc, ce n'est pas seulement moi qui travaille. Bien sûr, c'est mon laboratoire entier. Et j'ai choisi quelques thématiques dans mon laboratoire qui touchent vraiment l'application génomique au niveau des interactions entre les hôtes et les herbivores ou les pathogènes. Je travaille aussi euh, euh, de plus en plus sur les pathogènes. Mon laboratoire fait différentes choses aussi. Donc, je travaille aussi sur les peupliers plutôt. Que j'ai commencé déjà quand j'étais postdoc à UBC. Donc, je travaille sur la qualité du bois. Je travaille également maintenant sur la problématique du changement climatique et d'identifier les populations qui sont plus résilientes que d'autres, qui sont plus cibles d'avoir des impacts négatifs à cause des changements climatiques. Je travaille aussi sur, plus spécifiquement sur les peuplés faux-trembles et je suis en train de mettre en marche un genome browser pour aider les analyses fonctionnelles chez les peupliers faux-trembles. Et je travaille aussi sur un projet de phénotypage à haut débit avec les drones. Ça, c'est un projet qui est piloté par l'Université de Toronto. Et je collabore avec Ressources naturelles Canada et aussi le ministère des Forêts au Québec. Donc, ça, c'est une petite introduction. Et je vous remercie d'être là, très nombreux, pour cette lunch et pour m'écouter. Donc, j'ai décidé de faire ma présentation en anglais pour être plus inclusif aux autres gens qui ne sont pas très à l'aise avec le français. Donc, je vais continuer si ce n'est pas un problème avec anglais. Donc, ça, c'est ma petite présentation pour débuter. Et so I will continue in English because I would like to be a bit inclusive to certain people who cannot understand French that well, if it doesn't pose a problem. So my work that I will be presenting here is basically looking at the interaction of hosts, especially conifer hosts, and their associated pests. And pathogens too. I am working also on pathogens more and more. So I have a joint project with Louis Bernier looking at the oak wilt disease. So we have hired a PhD student with us, Léa Lebert from France. who will be working the next 4 years or so on identifying some genomic signatures of the interaction between this host pathogen system. And I forgot to mention, I'm working also on oak, of course. Actually, I've done my PhD on oak genetics. And so with the help of my PhD student and also Lisa and my postdoc, Melanie, we are looking at oak genetics too. And this is in collaboration with Natalie Isabel from the Canadian Forest Service and other partners. So I want to start now with some work that we have done in different projects, basically on a huge genome project that is funded through the ELSA program from Genome Canada. And that was led by Richard Hamelin, and I was a co-lead here at Laval University. So the background of all this is really the crisis that has affected forests worldwide. So in Europe, where I come from, we have huge problems with spruce regeneration, actually. And Norway spruce is actually the major tree species in Austria for forestry. So in Canada, there's also a problem, of course, with drought, with increased fires, but also the work that I'm very much interested in is to look at the interaction of native or invasive pathogens and pests. So there is, based on climate change, there is a huge amount of incursions or host jumps, also of local pests that affect the forest. You can think about the mountain pine beetle epidemic in Western Canada that has spread. And then, of course, we have the problem that we have with invasive species. So as we speak, we can imagine that a new species has been introduced through globalization, through transport, and as a hideaway. And this invasive species costs a lot of money, especially also in the urban context. So this is an example. Oops, sorry. An example, the emerald ash borer, which is estimated to cost around 890 million Canadian dollars to replace actually decimated ash based on the emerald ash borer epidemic. Actually, we have that, as you know, in Quebec City as well. So actually close to my property, last year we had to cut down a tree based on this. So my interest actually to look at this pathogen or insect and host interaction stems from my first postdoc that I did out in British Columbia. So in this project, which was also funded by Genome Canada, we were looking at the interior spruce. The interior spruce that is growing in British Columbia, and is also an important plantation species for the forest industry, has been affected by a local pest, which is Pysodus strobi, which is the white pine weevil or spruce shoot weevil, which is affecting actually the upper part of the tree. So this is a healthy looking tree, obviously. But if this pest is actually affecting the tree, we would have rather trees which are very shrubby, very bushy, very small. And this, of course, is not merchantable wood anymore into the future. So here on this picture, you see actually Barry Jakish, which has been now retired, the tree breeder for interior spruce, and with whom I have collaborated very well out in British Columbia. So he provided us access to the spruce trial, to the weevil trial, which was established in 1995 already. And the way that the screening was done was they have actually in fall, in autumn of 1995, 1999, they have augmented the local weevil population that was already there. And then they have actually in the year 2000, so following, they have actually taken note and estimated the attack. So they had a certain way of noting down the severity from non-attacked to attack and to death of the leader. So that part that was really killed. And they also actually counted egg plaques up there in the leader. And this was done for about, I think, 4,000 trees, if I remember correctly, that was actually planted in this plot. So this is the outline of the plot. So you have the rows and columns. And this was arranged in a 3-block design. So, and the families were split between the 3 blocks. So I think there were 42 families in total that were screened. And the way that these families were generated was they crossed basically trees that were termed resistant based on previous knowledge with other resistant trees and generated a family. And then they also interbred susceptible trees with susceptible trees and also resistant trees with susceptible trees to generate in total 42 families, of which the majority was intermediately resistant, so resistant by susceptible crosses. The rest was resistant by resistant crosses, and the other part was susceptible by susceptible crosses. And they also measured the height data before and after the attack. So for us, for our analysis, we were more interested to look at the height data before the attack happened, to be able to make an inference if anything that is related to growth is actually affecting somehow the attack on the leader, on the tree. So we were looking basically at the relationship between growth and attack from the insect pest. So we wanted to know if there is any trade-off between growth and resistance, so to say. So our objective was actually to look at constitutive resistance, so basically preformed resistance that already existed in the tree. Another part of the project was dealing with induced resistance, so where they measured basically the newly formed terpenates, for example, or phenolics, some of the most important resistance compounds or defense compounds. So we used at the time microarrays to do transcriptomics, and we generated correlation networks between the differentially expressed genes that we have identified in 4 distinct families that were actually segregating for resistance and for growth very well, because they were based on crosses between a resistant and susceptible parent. And we actually identified one gene, which was the top gene for us in all these interactions, which we found as an ABC transporter. ABC transporters are huge gene families, and definitely they are huge also in conifers. So my former PhD student, Anthony, who is here as well, he was able actually to map this ABC transporter sequence to a good collection of ABC transporter genes that he generated the tree from Arabidopsis, I think poplar too, and then some other known conifers such as Picea glauca or Picea abies, so Norway spruce and white spruce sequences. And it turned out actually that this ABC transporter is mapping to a clade here that is really specific for conifers. And it does not map to this clade here where the blast results actually pointed to blasting against Arabidopsis, because actually in this clade, so far, not a single conifer sequence exists. So this pointed to us, and this is what I wanted to confer here as well, that working with conifers is pretty challenging because they are quite distant from the model species Arabidopsis. So it will be always important, if you want to go any further with functional characterization, to actually know the relationship to other sequences. So we cannot say that, for example, the blast result will give us some indication of autology at all. So this complicates some of the work, but we found that this would be a really good candidate to eventually do functional characterization of this gene eventually. So to do transgenic work in conifers or in spruce is also challenging. So we'll see where we will get there further. Within the ELSA project, BioSafe, which I mentioned before, we did like a spin-off project, a small spin-off project, where we wanted actually to test the potential to screen 10 host species, 10 conifer host species, potential host species, for the invasive Lymantria dispa-asiatica insect, which is a Lepidoptera. A Lepidoptera similar to the spruce bartworm, also Lepidoptera species that you're familiar with perhaps. And this was one of 4 species that we chose for this ELSA project BioSafe to look further into. We selected it because it has several interesting characteristics, different to the already established European sister species that you can see here in red. That was in the 19th century accidentally introduced from Europe, actually, where it is native. That's the native range of the European subspecies. The difference between the European Lymantria dispa, so it's called actually spongy moth. Previously it was called gypsy moth, but the name was changed a few years ago into spongy moth, is that the female individuals actually shown the range here in green of the Asiatica sister species is actually flight capable. So the male individuals are always flight capable, whereas the female individuals are only flight capable in the Asiatica, so in the Asian sister species of the spongy moth, and they are not flight capable for the European one. Also, it was found that the Asian sister species is attacking actually more host species than the European sister species. It is not established in Canada, but it is one of the high alert invasive insect species, insect pests. So the idea was here actually to identify the potential of susceptibility in 10 conifer hosts that are shown here of different genera. So we had lodgepole pine, so we had spine, we have tested lodgepole pine, we have tested white pine, coastal Douglas fir was interesting to us, tamarack, eastern hemlock, balsam fir, black spruce, Norway spruce, white spruce, and western red cedar. So Norway spruce was chosen. It is, of course, an exotic species, which is native to Europe, but it is actually an important plantation species here in eastern Canada, in Quebec. And then we had also an oak species, black oak, Quercus velutina, which is the preferred host for this species. So what we did is we did a feeding experiment, which was a non-choice feeding. So the insects didn't have the choice to choose among the different hosts, but they had to basically eat what was given to them. And they were in all in separate containers depending on the host. And we also took material, leaf material, prior to the feeding to actually test for some constitutive defense compounds, in this case, terpenoids and phenolics. So we did GC and LCMS analysis. And then we compared the success rate of the larvae, so how well the larvae actually did upon feeding. And in terms of survival, in terms of weight gain, and in terms of development, up to the day 10, so which instars they were able to develop to. And we did also a transcriptomic experiment because we isolated at the end of the experiment actually the midgut from the larvae and looked for differentially expressed genes there. And we tried to combine all this information to give us some indication, first of all, which among those species is the most susceptible and also resistant, and also how actually could we interpret the development of the larvae in terms of the content, the chemical content of the needles that we have here in the different hosts, and also how the larvae is responding in terms of changes in expression in the intestinal tract. So once we have done that, we knew which species, at least from those nursery plants that we were using, seems to be most susceptible and which one most resistant. So it turned out, and I will come back to that later, that Norway spruce is actually the most resistant species, which is the exotic species, and is actually found here. It's this distribution. So in the further east, it will hybridize with Siberian spruce, but it still exists here in the east. So this is not surprising to us if you think about some sort of coevolution between this species, the sister species that comes from here, from the east, and the host, right? The distribution of the host, where the host goes. So there seems to be something going on there. And also not surprisingly, the species that we tested here from the west coast of Canada, the coastal Douglas fir, we found as the most susceptible species. There was also actually a lodgeable pine, relatively susceptible too, but we embarked actually at the two extremes, like the Douglas fir species and the Norway spruce species for further analysis. So we decided to focus on the terpenes here for technical reasons, for interest reasons in terms of constitutive compounds. So we had access, thanks to the Canadian Forest Service, of 5 provenances from Norway spruce that originate here from as far east as possible, including Belarus, Lithuania, and Poland, I think, as well. So relatively east, as east we can get with the provenances that we have planted here in Quebec. And then we got access to the breeding trial here in British Columbia. So we were able to have access to the Douglas fir breeding trial thanks to the BC Minister of Forests, and we investigated 17 families of coastal Douglas fir for constitutive compounds of terpenoids. And we did statistical analysis to see, first of all, the diversity of the different terpenoids. So we did individual terpenoid analysis based on previous literature. So we had authentic standards that we could use compare this to our to the content in the needles of those two species, so Norway spruce and coastal Douglas fir. So at outcomes, we will see the chemical diversity that we find in those foliages from those hosts. We can interpret what we found to be the coevolution between host and pest, or the lack of coevolution, depending. And we are also able, because we have used controlled crosses, to estimate genetic parameters such as genetic correlations or heritability. I have to admit our sample set was not extremely large, but it was still good enough to give us some indication on the genetic parameters. So what we actually found here, we have, maybe I should have introduced this. So we have actually tested two different populations from the Asian spongy mouse, Limantra dispa, out far in the east, Harbin and Beijing. And so we compared these two colonies that were grown actually, or they were kept maintained in Connecticut at Ansonia at the USDA, or the US Forest Service. Our collaborator there was Melody Kina. And we found that, so as I mentioned before, that coastal Douglas fir, actually the larvae did best on coastal Douglas fir in terms of larval weight gain, as you can see here. Also in terms of development on the instars. And so it even went up to the 4th instar within 10 days, what we couldn't find at all here in Norway spruce, which you see here completely on the left in both cases. And of course, it is sort of intermediate with the preferred host, which you see here, which is the black spruce, where the larvae developed the best as expecting. So we looked at the diversity of the compounds. Here you can see the terpenoids in the nursery plants that we used for insect feeding. So we identified actually 3 clusters in terms of the chemical diversity among all these species here, these conifer species, and also oak, which is shown here. And we also found a significant correlation actually with the development of the larvae, or any information that comes from the differentially expressed genes in the larval midgut that we harvested for 9 terpenes. And I want to point your attention especially on the camphene bornyl acetate and the isomer alpha-pinene. And also we found this significant correlation for 2 phytohormones, 12 flavonoids, 6 phenolic acids, and 3 lignans, and others as well. So we decided to look more in adult trees then. So we wanted to compare the nursery trees, the foliage from the nursery trees with the foliage from adult trees. So this is a picture taken apparently from the establishment in 2003 from the Douglas fir progeny test. Unfortunately, I don't have another image, just this one that I found. But the trees were obviously much older when we harvested them in 2020. So basically, based on our analysis, we found that these families, the 17 families that we used for Douglas fir, grouped into 2 distinct clusters based on the chemical diversity in the foliage, and this is based solely on terpenoids, as I mentioned before. We also found, because we were interested to look at the difference between the chemistry of the nursery plants and these adult Douglas fir trees, and focusing only here now on those three terpenes that I mentioned before, because we found them interesting in terms of correlation with larval development or anything related to gene enrichment from the larval midgut when they were feeding on the foliage. We found basically that for camphene and alpha-pinene, we found higher content in needles of the adult Douglas fir, and this was true for the 17 families. But for bornyl acetate, we found no difference. So bornyl acetate, actually, camphene as well, were the two most important terpenes that we found overall in the foliage of the conifers. For Norway spruce provenances, we found only for camphene significantly higher content on the adult Norway spruce trees as compared to the nursery trees. We didn't find any difference for bornyl acetate or alpha-pinene. And then we also looked now in Douglas fir on some of the genetic parameters. So Ebenezer calculated the genetic correlations among the tested terpenate compounds, and he also estimated the narrow sense heritability for those compounds. So among all the tested 32 terpenoids based on the content drawn from the authentic standards, where we could quantify actually those compounds, we found only two terpenes, the two sesquiterpenes, isolongifolane and aromatendrene. They showed a higher narrow sense heritability, so greater than 0.6. The rest was relatively low. But thanks to this network and to this knowledge, actually, we found that actually these two sesquiterpenes are genetically highly correlated with our three monoterpenes of interest, camphene, bornyl acetate, and alpha-pinene. So they show genetic correlations actually larger than 0.9, which actually points to the fact that we could select for these monoterpenes, which we found of interest, indirectly through selecting for those two sesquiterpenes. Another thing that we were interested to look at in the BioSafe project is actually on the pest side to identify genetic markers that could be used in a surveillance tool. In a surveillance tool, tell us if we have intercepted unknown samples, where do they come from? So to do this, of course, we need a sample collection, and this has to come from the native range. So in this case that I will be talking about, we were dealing with the Asian longhorn beetle, who is native to China and Korea. So we did an elaborate sample collection to have enough power to actually do some inferences about samples, about specimens that occurred here in North America. So basically, we applied standard population genetics to give us some information about the native range of the Asian longhorn beetle. And here on the left side, you can actually see how many incursions and ongoing, I would say, occurred actually starting from the mid-1990s. So started out in New York and is continued further. And I think the last one was in Southern Carolina, I think. And also in Europe. So the Asian longhorn beetle is a really big problem because it's a wood-boring insect, and especially for Canada and also the the northeastern United States, the sugar maple is actually a preferred host. So how this all became actually an invasive pest? So it is assumed that the problem started because it was a non-conspicuous individual actually before, but it became a pest. Even in its native range, it became a pest. The reason is there was a huge or still plantation here in the north of China to actually fix the desertification to the Gobi Desert. So they planted a lot of poplar species, and somehow what happened is the insects of the Asian longhorn beetle really expanded hugely, massively, and was moved around based on wood packaging material. And so it became basically a huge pest also in Europe. And it also occurred in North America and was in certain places was eradicated. In other places, it's still lingering. So my postdoc, former PhD student Ming, has done all the population genetics work. She published a paper in 2022 on the population genetics in the native range of the Asian longhorn beetle. And in certain parts, like here, the population structure is pretty complex, actually. But she found the potential to use a certain amount or minimal amount of markers, actually, to assign individuals with relatively high accuracy, over 90%, either to a north-south gradient of populations or even through 6 different regions. Of course, the more you are increasing basically the resolution, the more markers you would need. So you need, as you can see here, more markers to assign to those regions than just assigning to this north-south like gradient. And then, based on this knowledge that we have from the native range of the Asian longhorn beetle, she was also looking in the invasive range here, only North America. So there were samples from New York, in Illinois, of course, Ontario, Toronto was very important to us to figure out where the origin from this incursion came from. Massachusetts, we didn't include the last sample here from South Carolina, though. So the idea was here to see where basically the incurred or the intercepted samples originated from. So based on the analysis that were done previously on the structure in the native range, Ming was able actually to assign with relatively high confidence most of the incursions in North America to a certain region. So in this case, as you can see, most of the incursions seem to come from this region, actually, from North America, including from Toronto. We were also looking or interested to see if there has been some selection ongoing in the native range. So this is an example of a gene which is included in cold cardinals in the Asian longhorn beetle, glycerol kinase. And this pattern shows you actually a relatively nice alignment with the decrease in temperature. So I have another slide here showing you the temperature distribution. So here, of course, it's much colder than here. And if you overlay this with this pattern here, you can see actually the increase of this allele A, the colder it gets. So this is a nice corroboration actually of the findings that she had. Another thing that's interesting, but this might be known to people who have actually worked on the genome of the Asian longhorn beetle. The Asian longhorn beetle is genomically or genetically very much related to the mountain pine beetle, which is actually a native pest here in Canada. And so this gene, which is here designated as a hypothetical protein of Dendroctonus ponderosa, which is the mountain pine beetle, is actually closely related to the gene from the Asian longhorn beetle. So this is also a nice confirmatory study. We have also been looking not only on SNPs that come from GBS data. This is what we have actually used for the population study, and it was done in-house based on the Ion Torrent platform. But we have also done whole genome sequencing, and to stay cost effective, we used an approach called pool-seq. And for this, we generated 10 pools of DNA sequences, 10 new populations, so to say, where those pools were based on some genetic similarity, but we eliminated all highly related individuals in these pools, and also based on the climate, actually on climate data. So we generated 10 pools, and each had 10 individual DNA specimen, basically. So we did, or Mink did outlier scans, and she here in gray, she identified more than 70,000 outlier SNPs, which could be designated to over 5,000 genes. And she found 5 different categories for CAG enriched. So like fatty acid biosynthesis, TCA cycle, glycolysis or glyconeogenesis, fatty acid degradation, fructose and mannose metabolism. So, and those are all somehow related to energy metabolism. And then she also looked at the associations of genes specifically with climate data. So she tested 2 climate parameters, first mean temperature of coldest month and isothermality. She found GO-term enriched results, but no CAG enrichment was found here. And finally, a last thing that I want to mention, where I've been involved in, and which is a collaborative project actually with the University of Chicoutimi, but we have also here a like a collaborator and an entomologist at our department, Eric Boos, is actually to fight the spruce bartworm with its own DNA. So the rationale behind this, and this was actually work that has been initiated at the University of Naples in Italy, was to use the concept of extracellular self-DNA to somehow induce an immune response that is then based on a high concentration toxic to the insect. So as you might know, the spruce bartworm is actually, the major fight against the spruce bartworm is actually spraying PTK. PTK is from a protein of Bacillus thuringiensis, which becomes toxic when ingested by the spruce bartworm. So this is usually used and sprayed on the trees to somehow diminish or get rid of the spruce bartworm. So it does have several disadvantages. One of the things could be also, there could be some sort of resistance induced. But the idea here was to use this extracellular self-DNA actually to induce immune response. So for this kind of project, the idea was to produce fragmented DNA, and then to produce a lot, and then encapsulate this, and then perform toxicity tests on the larvae, as you can see. And we are still working on this, and we have done some toxicity tests. And I think there's Sirin Bubaker whose work that is, and she is continuing those tests here at the Faculty of Forestry to see how well this goes. And so the main lead of this project is Annie Deslauriers from the University in Quebec and Chicoutimi. So I will stop here, and I'm apologizing for this old picture. I have been on sabbatical leave, and I have to update my lab image. But I would like to mention, though the financial support, the continuous financial support from Genome Canada, Genome Quebec, and CERC, also the Max Planck Institute, so Jonathan Gershonson's lab, and Almut Hammerbacher, who was so nice to provide actually free of charge the phenolic analysis for the foliage for our nursery plants, where we did the insect feeding. The continuous support by Natural Resources Canada, the Canadian Food Inspection Agency, and the FRQNT, and the Ministry of Forests in Quebec, and also British Columbia. Merci, et je prends les questions en français ou en anglais.